Welcome back, everyone. It is now time for Lars Wahlberg and Sinfonia Biotherapeutics. A warm welcome, Lars. We're looking forward to seeing your presentation.
Well, thank you for having me. I'm sorry I can't be there physically, but I'm looking forward to the interaction with you and presenting Sinfonia. So Sinfonia is a biotherapeutics company that we're really looking at developing neurological treatments for great medical needs and not only symptoms, but also trying to repair the system. So I'll make an introduction to the company and then spend most of the time on what we're trying to accomplish with our lead indications and pipeline products, and then discuss a little bit our investment case and in perspective of relevant deals. So we're a privately held clinical stage company. We are a group of people with background in pharma and entrepreneurship, and we're treating an unmet medical need with a focus on a late-stage product in neuropathic pain and also other degenerative disorders that have a great medical need. We are using the power of evolutionary conserved proteins that are involved in the development of the nervous system, and when we can reapply them into the diseases, we can then not only treat the symptoms, but also have reparative type of effects. We have a portfolio of high-value pipeline at various stages of development. The late-stage late-stage product is in a new repair, and it's already completed a phase 2A proof of concept in the clinic, and we're looking to continue that now in additional clinical trials and indications. We have a preclinical development stage gene therapy product for pharmacoresistant temporal lobe epilepsy using our proprietary gene therapy platform. And that platform can also be applied in Parkinson's disease, which we have a research program on. We also have a research program in an orphan disorder called frontotemporal lobe dementia. Our business model is really to partner the product pipeline really at any stage of development if the deal is right. But obviously the phase 2 product is the one first up. And we're looking for a market authorization with a partner then in 2026. We are a dedicated core team of with backgrounds both in medicine and in science. And myself, I'm a neurosurgeon by background, but have been in the industry for more than 20 years. And my co-founder, Johan Lundqvist, comes from neuroscience at the Karolinska, and then from AstraZeneca, and also working in entrepreneurship and new smaller companies. We have clinical development important for our phase 2 translation. We're covering the finance with Christopher Svensson. We have corporate development here in the US with Bob Blomqvist, and we have our chairman, Thomas Pollare, who's not only a doctor and has a background in pharma, but also worked in investment industry. So we're covering our basis here to build a good organization. Next slide. So we're targeting the nervous system, so both the peripheral and the brain system. So for the neuropathic pain, we give it systemically to treat the peripheral nerves, and we're targeting the pain in chronic sciatica. This is usually caused by a herniated disc in the lumbar region with severe pain that develops also chronically in many patients. So large market. Other injuries to the peripheral nervous system is through painful diabetic neuropathy, or diabetes causes pain in because of metabolic disturbances as well, and that's also a huge indication. Where we need to go more directly into the nervous system, we apply then more of a direct approach with our gene therapy device, and we're targeting the pharmacoresistant temporal lobe epilepsy, where we can go directly into the temporal lobe to treat epilepsy that doesn't respond to normal treatment. And that's also a large number of patients. And Parkinson's disease, even though there are several treatments around the corner, it really doesn't target it the way we do. And we also go after an orphan with frontotemporal lobe dementia. So broad portfolio of products going forward. Next slide, sorry. So the pipeline and timeline, we are already completed a phase 2 with our new repair product. And we have clinical supply ready, and we have lined up the CROs. So we are ready to file an IND at the end of this year, and then go into phase 2B in 200 patients in sciatic pain, and start a new indication with diabetes in 100 patients in phase 2A. Top line results from the diabetes we expect next year, and in the sciatica at the end of 2023. And as I said, we're looking at partnering those for further commercialization and further clinical development and approval. Our Epi Repair product, it'll go into preclinical development over the coming months, and then an IND will be filed at the end of next year, and we'll do a clinical trial at the Karolinska in phase 1B. And with that data, we expect that we also are able to do early partnering on our Parkinson product and our FTD research products. The new repair is based on an injectable formulation of a protein called Neuroblastine. It's a drug that then targets the sensory degeneration and the pain in sciatic and diabetic neuropathy. And it actually restores the function and regenerates and repairs those sensory neurons. And the market here is quite large. There are no current approved therapies for sciatica, and there are a lot of off-label use of pregabalin, antidepressant, and other things. So we are targeting a market with huge potential of annual sales, and same thing with in painful diabetic neuropathy, where current pharmaceuticals, even though they're prescribed, are inadequate. And so there's a large need for new effective treatment. And in this particular case, we're also looking at the potential disease modification in the sense that we can also restore the sensory function and not only treat the pain. And why I'm saying that is that in animal models, our molecule really can regenerate the nerve fibers affected in disease. And this is a study done in rats with our collaborator in Frank Pareca at University of Arizona, where they did a mechanical injury to the nerve roots. You can see those red X's from the cervical region all the way to the thoracic, basically impairing the sensory function in the left forelimb. And if you look at the lower figure, that creates a permanent sensory loss. So the yellow line above is rats treated with vehicle, and so you don't get any improvement. But if you look at the blue line, the animals treated with a sub-Q injection of Neuroblastine then recover after just 2 weeks of treatment, and then they stay recovered for several months. And if you look at the video, I'm not sure how I start the video here. I guess maybe I can't. Yeah, there we go. Thank you. And if you look at the vehicle treated, they have at 6 months a permanent damage. And if you look at the lower video, if you could start that for me, please, you can see that those treated animals have normal function. So this is very powerful. These nerve fibers, if you look at it histologically, actually go back into the spinal cord and reconnect to it. Similarly, in the right figure there, it says name A. This was also done in a model of pain, basically in the lumbar region where you would normally have a herniated disc in humans. And you can see here that we're also restoring both the sensation, but also alleviating the pain. So we have a large amount of preclinical data that supported the clinical translation. So this was done in collaboration with Biogen in the US, where patients with sciatic pain, chronic sciatic pain, got 3 doses of Neuroblastine for 1 week. And you can see in the right-hand figure, there were doses ranging between 50 to 1,200 micrograms per kilogram. And unfortunately, the adaptive design that they selected, the computer randomized the patients to very few in the intermediate dosing. But if you look at the 50 and 1,200 microgram that had significant number of patients, you can see then the significant pain reduction in what's called average general pain intensity. So this is a scale normally used for pain assessment. And you can see the lower figure, the Neuroblastine at 50 micrograms per kilogram, then crossed a significant effect in the order of the same pain relief that Lyrica in painful diabetic neuropathy was used to register for. And that's one of the best-selling brands in neurology with $3.3 billion sales in 2019. And this effect then lasts for the following 3 and 5 weeks. So for the gene therapy device, then, which are in preclinical development for epilepsy, this is a new platform that we can apply to treat diseases of the brain and central nervous system. And it builds really on something that's already established, where deep brain stimulation is currently commercial product. Neurosurgical Center has the equipment to implant these into the brain at very low risk, minimally invasive. And if you show the video on the left, what is done is basically a small burr hole where these devices go in. And so our devices then go into the brain, and instead of delivering electricity like an electrode, can deliver these regenerative proteins locally within the brain, and then have an effect of gene therapy. The nice thing about it is that you can then remove these devices, you can replace them, and unlike in vivo gene therapy, where you actually modify the patient's own cells, we don't do that, and have a huge safety advantage and ability to actually manufacturing a device outside of the body and do all the testing before putting it in. The device concept then is that we have a device that is secured at the burr hole level, and the active region is placed in the target using this, what we call tether in blue there. It's basically just a handle. And then the tip consists of a polymeric membrane that's semi-permeable, and on the inside we have a human cell line that we can engineer to secrete factors, and also it survives inside. And we've already been in the clinic with this for a pilot in Alzheimer's disease at the Karolinska, showing that we can implant these safely, we can remove them 6 months and a year afterwards and safely, and they're still secrete. Similarly, we have a sister company doing the same technology, but for the eye, and they have already had patients implanted for 5.5 years, and the device are still functioning. So we have a pathway from a regulatory pathway established, we have the manufacturing established, and we have got strong clinical partners at the Karolinska to support the translation. So we're going now into the epilepsy. We have strong data in a couple of different animal models to treat the pharmacoresistant epilepsy through placement in the temporal lobe. And this is the normal procedure that the neurosurgeons are well versed with. And you can see the data there in the columns of the data, showing the control in white and the treated in blue. And you can see over time in this model that we reduce the epilepsy. And interestingly, if we pull out the device, I'm not sure you can see it, but there's a little arrow pointing down after 90 days. We actually get some epilepsy back, but not to the level of the control animal. So we seem to have also a disease-modifying effect of these implants. Similarly, we have a program in Parkinson's disease where we can then put our devices also in a different area and combine factors targeting both the lysosomal dysfunction and also repair the synapses with a different factor. And this then can create a whole new paradigm really for the treatment of Parkinson's disease. Unlike in vivo gene therapy, we don't modify the patient's own cells. So the nice thing is also that the pharma has gotten their eyes open for these types of treatments, new proteins, not just antibodies and small molecule drugs, and a large number of deals have been made lately on these new types of treatments, and we're already talking with some of our potential partners. So we are targeting a huge market with a competitive and an innovative portfolio. We have already a clinical stage therapeutic that is under GMP storage right now that we can go forward with very quickly into additional clinical study to partner and have therefore a de-risked late-stage candidate. And we are creating a nice pipeline at different stages with high-value disease-modifying precision therapies. So with that, I thank you for your attention and look forward to a discussion.
Thank you very much, Lars, for a very interesting presentation. I would like to start with asking if you could explain Neuroblastine's unique pain-relieving mechanism and how it differentiates itself from other drugs. And do you think it will have effect in other forms of neuropathy in sensory nerves?
Yeah, it's a very good question. The Neuroblastine is a non-opioid, so that's a great news for these patients suffering from pain. This type of pain is neuropathic, meaning that the nerve has been injured, so it's not the normal pain that you feel by pricking yourself with a needle or something like that. And the Neuroblastine then has this receptor specifically on the sensory neurons. And when you look at the markers of neuropathic pain and so forth, the Neuroblastine molecule actually restores it. And so it normalizes the damaged neurons. And when you look at then other potential indications of this is that any kind of nerve injury, so we have huge amount of animal data to support that even if you have a traumatic nerve injury or it's being cut off and so forth, even giving a systemic injection of Neuroblastine will actually regenerate that nerve and reconnect it in the spinal cord. So that opens up other types of injuries if you have a traffic accident with a nerve injury or a laceration or that type of things. The clinical trials are more difficult for those, and so I think that we're starting the right place with the pain and the painful diabetic neuropathy and sciatica.
Great, thank you, Lars. Next question, and for what reason have you chosen to conduct the clinical programs in the US?
Well, we already have the phase 2A already done here. We have one single regulatory authority with the FDA. We've already been through that process, and we've already lined up 30 centers that have very similar way of evaluating things. So we'll do a quality study with a single authority, and then also have the huge market that is quite acceptable for the registration.